Okay, so here we go again. I know this is not the best way to do it, but it has such a great mic, so I really like it. So I'm going to go ahead with it. So chapter 19 is all about organic and biochemistry. Really, we want to talk about the four macromolecules, and those are your carbohydrates, your fats and lipids, your proteins, and your nucleic acids. Starting out with carbohydrates. So carbohydrates are anything that contains carbon, hydrogen, and oxygen linked together. These are your simple sugars, and they can combine to find more complex carbohydrates. They're used as your main energy source, used for structure and proteins, and essential part of your genes. Carbohydrates includes all sugars, starches, and cellulose, which is, of course, is the crunchy starch that is in plants. So whenever you hear a scientist talk about sugar, they'll use the word saccharide. Saccharide is another term for sugar, and it means carbohydrates. So you should get in your head that carbohydrates equals sugar, scientific name saccharide. One sugar molecule is monosaccharide, two disaccharide, and three sugar molecules are trisaccharides. If we make a larger molecule, it is called a polysaccharide. So right here is what you want to note, and it'll be on your worksheet where it'll ask you what is the monomer and polymer for a carbohydrate. Monomer means what's the one unit name, and polymer means what's the complex unit name or more than one unit. So the monomer for carbohydrates is monosaccharide, whereas the polymer for carbohydrates is polysaccharide. Yeah, losing train of thought. All right, so when you add sugars together like glucose and fructose, you end up with sucrose, and sucrose is known as our table sugar. And this is just showing this is how we actually make our polysaccharides. So that went quick, but the main thing about carbohydrates is that the scientific name for it is sugar. For sugars is saccharide, that the monosaccharide is the monomer, polysaccharide is the polymer, and that we make polymers or polysaccharides by adding two sugars together. Another thing I need to know is that all sugars end in OSE. So anytime you see that ending, you know that it's a sugar. So glucose, fructose, sucrose, maltodextrose, maltose, dextrose, sucralose, all of those names that you see on the back of your labels. Anything ending in OSE is a sugar. So the second macromolecule we want to talk about is proteins. And proteins, when you usually think of it, you're thinking, oh, she's talking about meat. No, proteins are actually polymers that are made of amino acids. So the monomer, or the small unit of a protein, is known as an amino acid. Please note this, circle it, highlight it, and write somewhere near that it's the monomer. Proteins include all of your enzymes in the body, hemoglobin, which is your blood, all of your hormones, and we know about those, and antibodies, which help your immune system. So amino acids have a certain look to them. They have this amino group here, having a nitrogen and two hydrogens, an acid group, the C double bond O, OH, and then this side chain R group. This R group is what makes amino acids different. You have 20 common amino acids, but you have different side chains for each of them. So amino acids linked together form what we know as polypeptides. And so when we talk about the polymer for a protein, it is a polypeptide. So the monomer for a protein is an amino acid. A polymer for the protein is a polypeptide. Forgive me, you know how it goes. All right. There are 4 levels of structure for proteins. You have your primary, secondary, tertiary, and quaternary, and you need to know how those look. Whereas primary is what we call beads on a string or pearls. You have secondary, there are 2 types. You have an alpha helix or a beta pleated sheet. And then tertiary is what I call a globular structure. And when I say globular, I mean something like a jello mold. You remember? Or maybe my grandma was telling somebody who made them. But when you have a jello, in the bunt pen. So that's a 3D tertiary structure. And if you put 4 of those together, you'll get the quaternary structure for proteins. So know that proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary, and what those structures look like. Your third macromolecule is nucleic acid, and this is one of the very important ones. So nucleic acid has a monomer known as a nucleotide. So the monomer for nucleic acid is a nucleotide. And you need to know the 3 parts of the nucleotide. You have a 5-carbon simple sugar. For us, the nucleic acid we have, the sugars are either ribose or deoxyribose. You have a phosphate group and a nitrogenous base. And there are 5 of the bases: adenine, guanine, thymine, cytosine, and uracil. So these monomers link together and they form the 2 nucleic acids, which are your polymers, DNA and RNA. All right, so this may have gone too fast. So let's rewind and go back. Nucleotides are the monomers for nucleic acids. I'll say it again. A nucleotide is the monomer for a nucleic acid. I then told you that a nucleotide has 3 parts. It has a sugar, a phosphate group, and a nitrogenous base. It could be either one of these 5. The polymer for a nucleic acid, there are 2 of them. You have DNA, which you've heard of before, and you have RNA, which I hope you've heard of before. DNA stands for deoxyribose nucleic acid. The sugar in DNA is deoxyribose. The bases are A, T, G, and C, meaning adenine, guanine, thymine, cytosine. I know I say them really fast. You can read them really fast or really slow, but you should know that the 4 bases for DNA are A, T, G, and C. RNA, the sugar, is ribose, and the R stands for ribonucleic acid. And you have 4 bases, A, U, G, and C, meaning you have adenine, uracil, guanine, and cytosine. Note here that there's a difference between DNA and RNA and the bases. Both have A, both have G, and both have C. The difference is RNA has U, which is uracil, whereas DNA has T, which is thymine. So if you were ever given a string of nucleic acids and asked if it was an RNA or a DNA, you can immediately tell because DNA would only have T's in it among the A, G, and C's. And then RNA would have U's in it among the A, G, C's. Okay? So that's important to note. So somewhere put the difference between DNA and RNA is that DNA has a thymine and RNA has a uracil. So talking more about DNA, DNA is composed of 2 strands known as a double helix. No one should leave a science class not knowing that DNA means a double helix. And if you hear laughing in the background, I'm at the Henry Center and the class is off the chain. Okay, so attached DNA has bases that are attached in between off the backbone. And so you'll find that it has a base pair rule that goes with this. So what is that rule? The rule says that in DNA, guanine always pairs with cytosine and adenine always pairs with thymine. So you'll often hear scientists or professors talk about it as ATGC. That means A always pairs with T and G always pairs with C. Now this particular slide is about 3 questions. It asks you guanine pairs with what, and you should be able to pick out cytosine. Adenine pairs with who, you should be able to pick out thymine. And then in RNA, adenine pairs with uracil. And so only in RNA does adenine pair with uracil, but G and C still pairs together in RNA. So do not get yourself confused. It is not hard. Just think clearly about it. In DNA, G pairs with C, A pairs with T. In RNA, G still pairs with C and A pairs with U. So you just want to make sure that you keep that clear in your head when you're answering the questions. So humans have 46 strands of DNA known as chromosomes. You get 23 from your mom and 23 from your dad. And so that gives us 23 pairs of 46 single chromosomes. RNA, we have 3 different types. RNA again stands for ribonucleic acid. You have messenger RNA, also known as mRNA, ribosomal RNA, known as rRNA, and then you have transfer RNA, known as tRNA. So messenger RNA is single-stranded, different from DNA, which is double-stranded. And then messenger RNA, as I said before, U pairs with A instead of it like DNA, T pairs with A. The good thing, or I shouldn't say the good thing, the most important thing to know about messenger RNA is that it is the RNA that's used to translate into protein molecule. Okay? So messenger RNA is the RNA that's used to make protein. You have rRNA or ribosomal RNA, and it is used to make ribosomes. And you have tRNA, and tRNA is specifically used for transferring specific amino acids to the ribosome for protein manufacture. So know what each RNA does. Messenger RNA used to make proteins, ribosomal RNA used to make ribosomes, and tRNA used to transfer specific amino acids. If you have those 3 things down and understand what they do, then you have 3 more questions that you answered correctly on the exam. And this just shows you, which everybody should know, DNA is used to make RNA, and of course it makes 3 types, tRNA, mRNA, and rRNA, which is made on the ribosome, and then these are used to make proteins. So tRNA transfers amino acids, messenger RNA goes to the ribosome where it meets up with the tRNA, and then in the ribosome they make proteins together. Okay? So hopefully that's clear. You know what DNA is, how the base pair rule works, which I've stated, and then RNA, the 3 types, and what they do. And the last macromolecule we're going to talk about are lipids. And lipids are non-polar molecules. And hopefully you remember a while ago, chapter 10, we talked about polar molecules, meaning water. And water we know is polar, and non-polar and polar don't mix. This is why oil and water doesn't mix, because oil is a fat and it's non-polar, so it floats on top of the water instead of mixing with it. So there are 3 types of fats: true fats, phospholipids, and steroids. And fats, as we know, can be saturated and unsaturated. So the difference between unsaturated and saturated, main difference is saturated has no carbon-carbon double bond, whereas unsaturated does have at least 1 carbon-carbon double bond. So what does that mean? If you did not remember it from our previous chapter when we talked about bonding and having a single, double, or triple bond, this is what we're talking about here. In unsaturated, you end up seeing double bonds or 2 lines, 4 electrons, whereas saturated has single bonds between the carbons. This picture, familiarize yourself with it because it does show up when we talk about fats. So the monomer for fat, the monomer for a fat is a glycerol and a fatty acid. So there are 2 things that make up the monomer. You'll have a glycerol and a fatty acid, and then the polymers for fats turn out to be true fats, phospholipids, and steroids. All right, so I'll repeat that so it's not confusing, because everything we talked about, we had a monomer and a polymer. And so for fats, the monomer for fat is glycerol and a fatty acid. The monomer for fats is a glycerol and a fatty acid. The polymers are true fats, phospholipids, and steroids. So this is the only one where we had 3 polymers. And the last thing from fats is to know saturated versus unsaturated. Saturated, single bonds between the carbons. unsaturated single bonds with double bonds interspersed between. All right. And we don't talk about that. So that ends chapter 19. Hopefully the quick and dirty of it you got and understand, and that should definitely help you with passing